Students, this photosynthesis. In the previous lecture, we have covered that the introductory part of the photosynthesis and then light reaction, is it? We have completed light reaction of photosynthesis, which takes place in the light reaction takes place in granothylakoid, is it? That is in the membrane system. In the membrane system of chloroplast. And what was the product of the light reaction? During the light reaction, there was absorption of light energy, is it? Absorption of light energy by which absorb the light energy? Who absorb? It is the photosynthetic pigments. By photosynthetic pigment, is it? Pigments, so that is chlorophyll and carotenoid. Photosynthetic pigment. Then there was the photolysis of water. In the photolysis of water, what was produced? That there was evolution of oxygen, then electron and proton, is it? That is hydrogen ion. These electrons were accepted by the chlorophyll reaction center of the chlorophyll, which before that, when it absorbs light energy, it release electron. So this loss of electron is made up by getting electron from water. And in this process, ultimately, we also get the ATP and the reduced NADP, that is NADPH plus H, is it? And this was the production of ATP and NADPH plus H. Okay, so this is called the reducing power or the assimilatory power, is it? This is assimilatory power. So there was a production of assimilatory power, and this occurred in non-cyclic photophosphorylation. This assimilatory power was produced in the non-cyclic photophosphorylation. So these were, so in the light reaction in brief, that is absorption of light energy, photolysis of water, and then the production of assimilatory power. This assimilatory power is utilized in the next phase of the photosynthesis, that what we call the dark reaction. Is it? This is called dark reaction. This dark reaction, the site of dark reaction is site, that is where it takes place. It is the stroma of the chloroplast. The site is the stroma of the chloroplast. Here it contains all the enzyme required for the dark reactions. And so here what happened, that the carbon dioxide is reduced, it is reduced to carbohydrate. Okay, simple carbohydrate, it is reduced glucose, fructose, and later on those glucose, fructose are converted into sucrose, and finally it is stored as a starch. So there is a reduction, and for the reduction, that assimilatory power, the NADPH2 and ATP are utilized. Okay.
So you're doing revision, right?
No, but that little bit of revision of this portion, that that is the what was the light reaction and what did we get from the light reaction. This portion was the revision, and now we are starting today. Our portion is the dark reaction. Okay, dark reaction of the photosynthesis. All right. In the previous class, we have not completed dark reaction. So today's topic is dark reaction. This one is the biosynthetic phase. This dark reaction is biosynthetic phase. This is called dark reaction because this is independent of light. Of okay, this directly, it does not depend on the light, it does not require light. Okay, so it can take place in the dark, in light, in both. But that light reaction, that was the light dependent process. Light reaction was, it was light dependent process. This is also, we call that it was the photochemical reaction. Actually, this is photo, that is light, photochemical reaction. Okay, and this dark reaction is the biosynthetic, and it is independent of light. Here what happens, that this is, as I told, the carbon dioxide is reduced to carbohydrate with the use of this assimilatory power which was produced in the light reaction. See, in the light reaction, the product was ATP and NADPH2. So this ATP and NADPH2 are utilized in the dark reaction for reduction of carbon dioxide. This dark reaction is also known as Kelvin cycle. Okay, this is also known as Kelvin cycle because that the biochemist, the Melvin Kelvin, Kelvin actually revealed the details of the reaction that takes place in reduction of carbon dioxide. Okay, so it was Melvin Kelvin who actually gave the detailed pathway through which the carbon dioxide is reduced to carbohydrate. Yeah, Melvin Kelvin was not alone. With him was his associates like uh, Benson. Benson was another associates of Kelvin, Benson and Boson. Okay, so it is sometime this is not only called Kelvin cycle, there is also known as Kelvin Benson cycle also. Kelvin Benson cycle, or it is also known as Kelvin Benson and Boson, means all the three names comes, for which this Kelvin and his associates they were honored with Nobel Prize. Okay, so this Kelvin, he actually used the unicellular green algae, that chlorella. Okay, chlorella is a green algae that belongs to chlorophyceae. This is unicellular algae. As I told before also that in the green algae and in higher plant the photosynthetic pigment constitution is same. So their mechanism of the photosynthesis is similar, and that's why most of the research works, experimental works were carried out in the green algae. Okay, so he used chlorella and he used the heavy isotope of carbon, that carbon 14, that heavy isotopes of carbon was used. Carbon, that he used the carbon dioxide that contain heavy isotope of carbon, carbon 14. Okay, normal carbon is normal isotope of carbon is 12, is it? And this one is heavy, so it is carbon 14. So carbon dioxide containing heavy isotope of carbon was supplied to chlorella to carry out photosynthesis. And using this method, he could detect the pathway, he could trace the pathway of the photosynthesis, uh, pathway of reduction of carbon dioxide. He also used the another method which is known as mm, chromatography. Okay, chromatography. This was, of course, he used paper chromatography. This is a method by which the from a mixture their component can be separated. Okay, say mixture of chemicals are there, and those chemical individual chemical can be separated through this method. And this chromatography, there is a simple chromatography, and of course he used the two-dimensional, that is 2D paper chromatography. Okay. And by using, he could trace the pathway of the carbon dioxide reduction. Another thing is that this Kelvin cycle is also known as C3 cycle. C3, C is carbon, carbon 3 cycle, and the plants where this cycle operates for the photosynthesis, those plants are known as C3 plants. So C3 cycle and C3 plants. Actually, why it is called C3 cycle? Because when the carbon dioxide is absorbed by the plant, the carbon dioxide is absorbed by the plant, then the first stable chemical compound formed, that is a 3 carbon compound. 3 carbon compound. This is the stable product after the carbon dioxide is absorbed. And this product, name of this product is phosphoglyceric acid. Phosphoglyceric acid. Acid, in short, we can say PGA. Okay. Sometimes it is also read as phosphoglycerate. Okay. This one is 3-phosphoglyceric acid or 3-phosphoglycerate. Phosphoglycerate. This three, of course, this is a 3 carbon compound, this phosphoglyceric acid. Okay. And this one, these three indicate that in that carbon compound, phosphorus is attached to where? Where is the phosphate group? Not only the phosphorus, phosphate group. Phosphate group is attached to the third carbon of the phosphoglyceric acid. So it is read as 3-phosphoglyceric acid. You can also simply can write phosphoglyceric acid or phosphoglycerate. Okay. As the first stable compound is a 3 carbon compound, so this cycle is also known as C3 cycle, and C3 cycle is the basic process for the reduction of carbon dioxide to produce carbohydrate. Now we will see in this is a more or less a cyclic process. Okay. The reaction, it is a chain of reactions, and that reactions are in a cyclic form. Cyclic chain of reaction. Okay. And the entire process we can at least divide into 3 main reactions. Okay. That is phases of the C3 cycle. This phases, first one is known as carboxylation. Carboxylation is the process by which carbon dioxide is absorbed and incorporated in a compound. All right. So it is carboxylation. Number two is, after that, it is the reduction. Reduction, this is the reduction of the phosphoglyceric acid. By carboxylation, this phosphoglyceric acid is produced, and next step, that the phosphoglyceric acid is reduced to another compound, and then there is, this process should continue. Okay, this reduction, this synthesis biosynthetic process should continue, and for that some compounds are always required. So those compounds should be generated again and again. And that's why this is the third phase, or the third main steps of the C3 cycle, is the regeneration.
So can you explain what is reduction again?
Yes,
So that reduction.
reduction is here actually reduction of phosphoglyceric acid, reduction of PGA to PGAL. PGAL is phosphoglyceraldehyde. Okay, PGAL is short form of phosphoglyceraldehyde. So here the after this by carboxylation, after the absorption of carbon dioxide, PGA is produced, and then PGA, phosphoglyceric acid, is reduced to phosphoglyceraldehyde. In this portion, in this reduction, that assimilatory power NADH2, NADPH2 and ATP are consumed. Okay, so this is what happened. Of course, I will show you the cyclic path. So now let's see only these things, this. After that, from the phosphoglyceraldehyde, carbohydrate is synthesized. Okay, and some of the molecules from the phosphoglyceraldehyde, they are utilized to produce the compound which initiate this cycle. Okay, initiation cycle, initiation compound. That compound is also regenerated from PGAL. Means PGAL, one side it give rise to carbohydrate, and in another side it produce that compound which will initiate the next cycle of reaction. All right. So these three are the main steps. Now come to the, see here I will show you. See here I will show you the Of course, a short form of this dark reaction, that is the Kelvin cycle. This is, as it is written here, simplified version of Kelvin cycle. Okay. Here we'll keep it aside. Okay. First thing is that there is one compound called ribulose bisphosphate. Phosphate. Okay. Ribulose bisphosphate. This is also known as RUBP. All right. This ribulose bisphosphate is the compound which initiates the process of dark reaction. This ribulose bisphosphate absorb carbon dioxide. Okay. So the acceptor of the carbon dioxide is RUBP. Is the acceptor of carbon dioxide. So this one is important. You have to always remember the compound. This RUBP accept the carbon dioxide, but in presence of some enzyme, and the name of the enzyme is RUBP carboxylase. So RUBP accept carbon dioxide in presence of RUBP carboxylase to produce two molecule of PGA, phosphoglyceric acid. RUBP is a 5 carbon compound. Remember this? RUBP is a 5 carbon compound and carbon dioxide is 1 carbon compound. When together they are 6 carbon compound. This 6 carbon compound, they split into two molecules, similar, same molecule. So these are 3 carbon compound, two molecule of 3 carbon compound phosphoglyceric acid is produced, and this is the carboxylation phase. Here you can see this. Okay, see, this is this is ribulose 1,5 bisphosphate. 1,5 means, if you can't remember, don't write this 1,5. 1,5 means that this phosphate group in the ribulose is attached with first carbon and fifth carbon. That's why it is 1,5. This is actually a biphosphate compound. Ribulose is a pentose. It is a 5 carbon compound, and one phosphate group is attached to the first number 1 carbon, and another phosphate group is attached to the 5th carbon. So this is ribulose 1,5 bisphosphate. This ribulose 1,5 bisphosphate, this one absorbing carbon dioxide. Carbon dioxide, it is not alone, that carbon dioxide plus water from the atmosphere it takes, and there is carboxylation. Here is the enzyme, that is RUBP carboxylase. Okay, this causes the carboxylation, and the product is 3-phosphoglycerate or 3-phosphoglyceric acid. So this is first reaction. Now one thing, that this RUBP carboxylase, this has a dual nature. No, that it is not only function as carboxylase, it also function as oxygenase. That is, when oxygen is enough oxygen is present in the atmosphere here, it is plant atmosphere, that is inside the plant. Okay, if the oxygen is more, carbon dioxide is less, then this compound, this enzyme will help to absorb oxygen, not the carbon dioxide. Means it prefer oxygen. Okay, when it will prefer means absorb oxygen, then photosynthesis will reduced, because for the photosynthesis carbon dioxide is required. So this enzyme sometimes function as carboxylase, sometimes function as oxygenase, and that's why the enzyme has been renamed as RUBP carboxylase oxygenase. It has a rename, and this is in short. Always to write and read very big name, it is difficult. So it is now known as Rubisco. That is ribulose bisphosphate carboxyoxygenase. So now you have to write here, instead of RUBP carboxylase, you can write Rubisco. All right. So catalyzed by the Rubisco enzyme, the carbon dioxide is absorbed, and RUBP is carboxylated to 3-phosphoglyceric acid. The next step is this 3-phosphoglyceric acid is reduced. There is a reduction, and in the process of reduction, this produce a triose phosphate compound. This triose phosphate compound, I told, what is that compound? This is PGA is converted to PGA Al phosphoglyceraldehyde, and during this it consumes ATP and NADPH2, and it forms ADP plus NADP. Okay, so it is this portion is the reduction. This is say triose. Here it is written as a triose phosphate. Triose means that say in the carbohydrate there are several types of carbohydrate. Carbohydrate are classified, grouped into monosaccharides, oligosaccharides, and polysaccharides. These monosaccharides are further divided into different types based on how many carbon atoms are there. So this start with 3 and goes up to 7. So the one which contain 3 carbon atom, they are called triose. If there are 4 carbon atom in a compound carbohydrate, then it is called tetrose. If there are 5 carbon, then it is called pentose. Pentose is, we have seen here also pentose is there, ribulose is a pentose, ribulose, ribose, they are pentose. And if there are 6 carbon, then it is called hexose. Hexa means 6, hexose. Hexose sugar is glucose, fructose, they are the common hexose sugar. Glucose, fructose, they are the common hexose sugar. Then there are heptose, which there is pseudoheptulase. Heptulase, pseudoheptulase are there. So this one is the 3 carbon, this triose phosphate compound is produced. Actually phosphoglyceraldehyde is produced, and another compound which is a isomer of phosphoglyceraldehyde, that is which is known as dihydroxyacetone phosphate, DIHAP. Okay, dihydroxyacetone phosphate. These two are the isomers, so they can change, they can convert. DIHAP can change into PGAL, PGAL can change to this, and these two compounds are then used for the synthesis of carbohydrate. Okay, so they are the main compound. These are, they are the triose. All right, and also from these two compounds, the RUBP, again ribulose phosphate, is produced. During this reduction, as I told, that ATP and NADPH2, which has been produced, which was produced in the light reaction, that is consumed here, and the enzyme here is triose phosphate dehydrogenase. They have not written the enzyme name. Enzyme here is triose phosphate dehydrogenase. So in the presence of triose phosphate dehydrogenase, this 3-phosphoglyceraldehyde, the 3-phosphoglyceric acid is converted into 3-phospho glyceraldehyde, PGL. From here, you can see that there is some chain of reaction from this triose phosphate, sucrose or starch is produced. All right. And some of the molecule of this triose phosphate, as I told, that dihydroxyacetone phosphate and phosphoglyceraldehyde, they are utilized for the generation of the RUBP. RUBP is generated, right? And during this generation, actually from here, one compound is produced. That compound name is RUMP, RUMP, ribulose monophosphate. First ribulose monophosphate is produced. This ribulose monophosphate is converted into RUBP, ribulose bisphosphate or ribulose biphosphate, or you can say ribulose diphosphate. RUDP also it is called. RUDP, ribulose diphosphate. Okay. And during this process, here it is monophosphate, one phosphate group, but this compound has two phosphate group. So one phosphate group will come from where? From somewhere one phosphate group has to be added, and this is the phosphate group is supplied by the ATP. And so it is ADP is produced. ATP molecule break here, phosphate group go with the RUMP to produce RUBP, and then ADP molecules are produced. So another ATP molecule is consumed. For each molecule of RUMP to convert into RUBP, one ATP molecule is required. Okay. And here also, for every molecule of PGA to convert into or to reduce into PGAL, one ATP and one NADPH2 molecules are spent. Okay. This one, say, in this process, the ribulose biphosphate, initially it is consumed, but again this ribulose biphosphate is regenerated. So we can say in the process of this dark reaction that ribulose bisphosphate is not spent. It has become a compound. It is a medium through which the carbon dioxide is incorporated and it forms the carbohydrate. Okay. So what happened? Carbon dioxide is, this is a one carbon compound. Single carbon is there. And glucose, what is the glucose formula? C6H12O6. Means here 6 carbon. So if from the carbon dioxide the glucose is to produce, then 6 molecule of carbon dioxide is required, is it? So for production of 1 molecule of glucose or fructose require 6 molecule of carbon dioxide, is it? So if 6 molecule of carbon dioxide here we take, then 6 molecule of RUBP is required. And for that, how many molecule of 3-phosphoglyceric acid will produce? This is 3 carbon compound. So here 12 molecule will produce, is it? And for this 12 molecule of phosphoglyceric acid to reduced require 12 ATP molecule and 12 NADPH2. Is it? That is required. So 12 ATP and 12 NADPH2. Then here for the regeneration also it is 6 molecule is regenerated. All these 6 RUBP is regenerated. Each one require for the regeneration of each require 1 ATP. So for the 6 molecule to regenerate require 6 ATP, is it? So total ATP how many? Total ATP will be here 6 another ATP molecules are required. So for the production of 1 molecule of glucose or fructose, glucose or fructose, that is this is called hexose sugar. Fructose or fructose require 18 ATP and 12 NADPH2 and 6 molecule of carbon dioxide. So for each molecule of glucose fructose to produce 6 molecule of carbon dioxide, 18 molecule of ATP and 12 molecule of NADPH2 are required. So means you can write this 6 carbon dioxide plus 18 ATP plus 12 NADPH2 per molecule of glucose. Say here. Now we will see it in other way. Say for 1 molecule of glucose require 6 molecule of carbon dioxide, 6 molecule of carbon dioxide to reduce 18 ATP, 12 NADPH2. Then for 1 molecule of carbon dioxide, how many require? For reduction of 1 molecule of carbon dioxide 80 sorry 3 ATP and 2 NADPH2 are required. Okay, so these are the things that you have to remember. Who is the primary acceptor of the carbon dioxide? What is the enzyme responsible for the carboxylation? What is the first stable compound formed? And then what is the next compound after the reduction? What are the enzymes here that you have to remember? Okay, any question? Hello, hello
Sir,
student. Yes,
so what is chromatography again?
yes, yes.
What is chromatography?
See, for you, this is no need to go in detail. This is a there are different types of process for the separation. In chemistry, you learn, no, that from a mixture, their component can be separated, and according to the what type of mixture it is it, what are the types of component it is, whether it is soluble in some solvent or not, whether the components are large or small. If it is very big component, you can even hand pick also you can do. If there are some magnetic one, you can separate by magnet. No, like that way, there are, when there is a very minute thing, some chemical compounds, some small molecules are there. According to their nature, they also can be separated by several different methods. Okay, physical methods are there, chemical methods are there. So among them, there is a process known as chromatography. When you will go to the chromatography in BSc, I think you have this some practical on this chromatography in the plus 11. Oh, now I have to see whether you have this practical or not. Through the chromatography, there was one practical separation of plant pigment through paper chromatography. Here actually what happened, this is just like a race, no? You take part in the race running, is it? Whatever type of, whether you are running, taking part in the race, or it is bike race, or it is motor car race, or it is F1 race, or it is whatever it is. In the race, what happened? All the competent ones, they start from the same point, say for the zero line, is it? Once they start running, then what happen? Someone has a less speed, someone run in high speed. Then what will happen after some time? Say if we give them 10 minutes or 5 minutes, according to time or even more, then what will happen? They, according to their speed, they will be in a different position, not in the same line. So this is more or less in the chromatography, similar things happen. In the chromatography, different one medium is taken. In the paper chromatography, paper is a medium, a stationary medium. One is a stationary medium and another is mobile, okay, which can move. So here what happened, have you seen, observed sometime that the blotting paper, blotting paper, if you just dip, touch one corner of it in water or any other liquid, it start absorbing that liquid. If it is a color liquid, the color liquid, color things also will come up gradually. It move. Okay. So this is what happened. So in this mixture of chemical is a solution is prepared in a solvent, suitable solvent, and the mixture what is to be separated, the component from the mixture to be separated, that are taken on the paper as a spot, small spot, and then its one margin is touched or dipped in that solvent. This paper, what will do? This paper will start absorbing this liquid, and when it is absorbing the liquid, the solvent is moving. This component also, that mixer component also will start moving along with the solvent, and their movement will depend on their size and molecular weight. Okay, so different molecule, different component will move at a different rate. Their rate of movement will be different. So after some time, what we'll find, that some of the component is here, the other one is here, say other one is here, someone maybe has moved very slow, so they will move at a different rate, so they can be separated. They will be separated. In the similar way that you have, I think, practical of separation of photosynthetic pigment. Synthetic pigment. In the photosynthetic pigment, there is the main pigment that chlorophyll A, chlorophyll B, carotene, and xanthophylls are there. Here in a petroleum ether, okay, a solvent is taken, and on the blotting paper, we call them chromatography paper. On that paper, the extract of the plant pigments are spotted on the blotting paper somewhere, this chromatography paper, and then it is dipped in a solvent kept in one jar. The jar is closed, okay, it is left for some time, then this solvent start moving upward, and from here the photosynthetic pigment chlorophyll A, B, carotene, xanthophyll, they also start moving. In the process, this carotene move very fast. It is nearly, it start moving with the solvent front, with the solvent rate is very fast, and then in between is xanthophyll and chlorophyll AB we get. AB is lower. So all the four different color pigment we can see on this paper. So this way they can be separated. So there are different method in paper chromatography. Also there are different types of paper chromatography, and for different types of chemical that is present in the mixture, for them also solvent etc. is differently prepared. Okay, this is a method of separation of chemical compounds from a mixture. Okay, you will get some in some other books also you will get this C3 cycle or Kelvin cycle in detail. Okay, C3 cycle or Kelvin cycle in detail. That is the very large different compounds are there, little complicated one, but for you here it is the with the main reaction it has been shown. That's why it is a simplified version. Okay, anything else you want to ask?
No, sir.
No, you can say here that in your book here it is written see, say there is carboxylation, no, the Kelvin cycle. You can see this. There is this Kelvin cycle. There's a carboxylation is there. Then this cycle has been given. After that, this is a reduction. And the reduction, the shape, then regeneration is there. So everything is given. Right here is in out, that how many what is in the process. In the process 6 carbon dioxide is in and 1 glucose molecule getting. 18 ATP is spent and 18 ADP is produced. 12 NADPH is consumed and 12 NADP is produced. So this is C3 pathway. In the next lecture, I will explain you C4 pathway. All right. Okay. You have next class also, so I will stop it here. All right.